I'm going to give, over these first 2 lectures, if you like, the basics, okay? The aim of these first 2 lectures is that afterwards, you can understand what everybody else is going to say, okay? So, at this point, there's no stupid questions, okay? Just shout out. And this is the same for the people who are online. If you have a problem, just unmute yourself and talk into the microphone and say, excuse me, I don't understand. Okay, please, it's important. So, first thing we're going to be talking about is the language of epigenetics. So, I always start a training like this with some very simple discussion questions. What makes one cell type different from another cell type, muscle cells against a liver cell, given that they have the same DNA? I mean, they have the same DNA. All our cells have the same DNA. Okay, what allows some genes to be expressed and others to not be expressed? And do you actually think you have any control yourself, consciously, subconsciously, as to where the genes are expressed? Now, shout out, what do you think? Just instinctively. I mean, all our cells are identical, they've got the same DNA. Go on, Lisa.
So I think different genes are translated to different proteins, which different enzymes that have different functions, and so in muscle cells are different than liver cells, different proteins define what the cell does.
Exactly. But, I mean, epigenetics is actually, if you like, the science underlying it. It's how some genes are switched on and other genes are switched off. Okay? So, all the cells start with the same DNA, and it actually comes down to the DNA, how it's packed inside a cell, which bits are accessible, which bits are not accessible, which proteins can then bind to it, which then makes the cells different. And what makes the cell different is the genes that are expressed in it. But what makes those genes expressed is its transcription. Yes, so this is transcription factors, proteins that bind to the DNA to drive it. But what makes the genes accessible and expressible is epigenetics. Okay, and do you think we've got actually any control over it? Who actually did the pre-reading? I sent 3 papers out. Okay, one of them was an old Nature Neuroscience paper. Did anybody read it? One of the 2 of you, doesn't matter.
I guess we do have, to a certain degree, control over our genes. It depends on environmental influences. So the paper that we read, it was especially about the maternal influence and how they take care of their infant. But I guess also during our life, we have to a certain degree influence over what we eat, how much stress we have. We exercise, and I guess that does influence our epigenetics and in the long term, which genes are switched on or off.
It's exactly that. And the way to think of it, genomics, is like rolling a dice. You've got it, you've not got it. Okay? Epigenetics, it's like a game of poker. You've got your cards in your hand. What's important is not the cards you've got, but it's what you do with them. Okay? That's what epigenetics is. Epigenetics is playing poker. Genetics is just rolling a dice, and that's what you've got. Okay? So, I always put this up, always ask a lot of questions. Okay? The only stupid question is the one you don't ask. Okay? So, I'm going to start off with just some words. I've got sort of 4 or 5 slides. Okay? Is everybody happy with the idea that if somebody says locus, a locus is just a region of the genome? Okay? So, when we start talking about P's and Q's, in British kids, if you're told to mind your P's and Q's, it means be polite. But P and Q are the chromosome. It's the short arm and the long arm. So, for example, when we talk about 11p15, this means chromosome 11, P, the short arm, on the 15th stripe. Okay? So, just keep in mind, when we talk about a locus, It's a small region on the genome, and we can always identify it, okay? And these are called cytogenetic bands, okay? Very simple. That's a locus. It's just a region of the genome, okay? So if anybody else in the rest of the 3 days talks about a locus, quite fine. It's just a region of the genome where there's a bunch of genes and stuff. Okay? Allele. Is everybody happy with the word allele? Because now we're getting more into genetics. Okay, we talked about a locus. A locus is a big region, okay? Give me a million base pairs. An allele is down much more fine detail, okay? Often it's down to a single nucleotide. And we talk about dominant and recessive, etc. Classical genetics, Mendelian genetics, Ps, okay? Epigenetics, we've got the same thing. We have an epi allele, okay? And an epi allele is just an alternative state of a gene that has an identical DNA sequence. That's important for everything we're going to be talking about for the next 3 days. Identical sequence. Epigenetics does not change the sequence of your DNA. Okay? That's important. Okay, I think we're all happy with words like genotype and phenotype. Okay, phenotype everybody's happy with. It's a state you can measure at the end. The genotype is the state of your genome that you can measure. So of course, what also do we have? We have an epigenotype. Some people call them an epitype. Okay, so that's just the state of epigenetics. Okay, everybody happy with these words so far? Okay, epigenome. I think everybody's going to be happy with that. Genetics, moving through to genomics. Okay, epigenotype. Well, we have a genotype which is just an allele. That's what we measure. Epigenotype is the same. But these are modifications. Everything we're going to be talking about is how the epigenetics is altering something at the end through gene expression. Okay? So we can have positive modifications. We have negative modifications. And all the positive one does, it increases gene expression, makes more of it. Or a negative one switches something off. Okay? But of course, we need to remember, like genetic changes, epigenetic changes, they can lead to inappropriate expression. Okay? We're not going to talk about it at all really over the next 3 days, but in cancer, a lot of oncogenes, which are genes that drive the cancer processes, they're epigenetically regulated. Okay? And we've got one of the papers that you're going to be doing in the first Journal Club, where we talk a little bit about this, and we see the difference between a lot of what we're going to talk about is environmental influences in cancer, which it's the same processes, but they're quite different scales. Okay? But for the epigenetics, genetics is switching on and off. You've got the mutation, you don't have the mutation. Epigenetics, it's like the dimmer switch. Want a bit more? Turn it around a bit. Want a bit less? Turn it down. If that's all that you remember from the epigenetics of the theory, okay, epigenetics is a sort of a quantitative modulation. It's not the on-off. Okay? Genetics is, you've got blue eyes, you've got brown eyes. If it was epigenetic, you could have every color from blue all the way through to brown. Okay? Keep that idea in your mind. Okay. I've got a slide here on gene expression. Is everybody happy with the idea that we go from DNA to RNA to protein, going through transcription, which is the process of the DNA to the RNA, and then translation, which is the RNA to protein. And both of those steps can be modulated. Both of those steps can have this dimmer switch to make more or less. Okay? Now, gene expression is controlled at various points in the sequence leading processes. Okay? So, I'm not going to go into the details. I think I've got one slide on it. Generally, in a gene, you've got a region upstream before the gene, and here you've got transcription factor binding sites, you've got CPG islands, you've got a bunch of complexity here. After that, you've got the part that is transcribed, the part that comes through to the messenger RNA. Okay, and then at the end, you've got the 3' flanking region. Now let's just go back. So gene expression is the control of various points in the sequence leading to protein synthesis. And you've got transcription, it's the synthesis of the RNA to the complementary to one strand of DNA, and then translation, which is then reading the messenger RNA and making the protein. Okay? And there are a bunch of other processes we're not going to really talk about, but in your first strand of RNA, we've got a bunch of irrelevant parts that come through that are then cut out, and there's modification processes here, and then afterwards we get the translation and post-translation modifications. So is everybody happy with that? That's good. So I was worried that everyone, yes?
Can I ask what the flanking region is?
So the flanking region is the regions at the edges.
Okay. Is it like the stop part for translation?
No. Here what we have is the whole gene. Okay, so the gene is quite big. If I take, for example, stress, the glucocorticoid receptor, the whole gene is 100 megabases long. Okay, so this goes from the promoter regions all the way through the CPG island, the different first exons, the rest of the gene through to the end, the alpha and the beta exons at the end. 100 megabases. The splicing, where all of these irrelevant parts are removed, brings it down to about 3 kilobases. Okay, and then you've really just got your exons, your useful parts at the end. And the flanking regions are in the genome, so they're this 100 megabases at the start, and they're sort of at the edges. Okay, and they're important because there's lots of sequences there that play a role in regulation of expression. Okay, often you find DNA methylation and epigenetic processes happening in the 5' flanking region and the 3' flanking region. Okay, this is part of the reason I put this slide up here, because people will be talking about promoters. So the promoter is just the region at the start, and it promotes the gene expression. Okay? It's what drives it, it's what pushes it. Okay? So we're going to, some people will talk about promoters. That's all it is. It's what drives the gene expression. Okay? Nobody's lost. That's good. Okay, and I wanted to just put this slide up because we need to move away our thoughts. Particularly I'm talking to biologists, okay? For 30 years, we've had the genomic revolution. Sequencing everything's going to solve all of our problems. But we need to be aware. If you look here, the monozygotic twin concordance rate, and each one of these is a disease. And this particular graph I found has age of onset of the analysis. But monozygotic twins, identical genome, identical in utero environment, cleft palate, average 10% concordance between monozygotic twins. Anybody know what's caused that? Midwives should have a good idea. It's lack of certain nutrients in utero. You don't have access to enough acid in our family, folic acid, etc. So that's one twin that was just below the amount that was needed. And the other was just above the amount that was needed. Okay? Type 2 diabetes, it's about 50% concordance. Okay? Has a strong family running. But when monozygotic twins are not concordant, it's not genetic. So there's something environmental and epigenetic in that. So we've got schizophrenia, Crohn's disease, type 1 diabetes, breast cancer. Breast cancer, which everybody says it's the BRCA1 gene. Monozygotic twins are only about 15% conformed. So everything that's going on there is actually epigenetics. And the more we study...
I'm sorry to interrupt you, but we have a comment that it is difficult to hear you when you move away from microphone.
Well, in that case, I'll move the microphone. Sorry, is that better? Is it better if I talk like that? Can somebody just give me a thumbs up or a
It's
yes?
excellent. Thank you so much.
Okay.
Yes, very good. Thank you.
Sorry about that. So if you do have any questions online, do not hesitate just to dive in. Okay, so is everybody happy with that? With the idea that we need to step away from genomics, genetics, and sequencing and start looking more holistically, environment and everything. Okay? Can you just move on to the next slide, please? Because okay. So I've just put up here for everybody a definition of epigenetics. So epigenetics refers to the study of changes in the regulation of gene activity and expression that are not dependent on gene DNA sequence. Has everybody really got that message now? That genetics is changing the gene. Epigenetics doesn't change the gene, but it changes what is done. It's the game of poker. Okay? So while epigenetics often refers to the study of single genes or sets of genes, epigenomics is more global analysis, the entire genome. Okay? Just more definitions.